So excited to have you here and have a chance to talk about some of this stuff. I'll let Asta go first, and then I have a bunch of questions to ask you.
Okay.
Yeah, Mike, feel free to just jump in as I
I will.
ask questions
I
as
will.
well. Yeah.
Yep.
Okay. Perfect. Yeah. So, Rich, I know you started talking about the history a bit, but let's just take it back. Maybe you can get started by telling us a bit about your background and how you got interested in bioelectricity in the first place.
Sure. Yeah, my background, I started out in physics, and I got my degree in bachelor's degree in physics, and then went to graduate school at Purdue. And in the physics department, they had professors give us a lecture about their work. It was a rotating thing, so we'd be able to hear all the professors what they're up to and decide if we want to work with them. And one of the professors that came on to do that was Lionel Jaffe, and he was actually in the Department of Biological Sciences, but he liked to go to the physics department because he found the students there were better and more appropriate for his interests. He told us about his work in measuring electrical currents in germinating seaweed eggs. And I found that fascinating. At that point, I had never taken any biology in my career, and to think that you can actually measure currents in living cells, I thought was fascinating. So I went and did a rotation with him, fell in love with the whole idea of measuring these currents, and I did my PhD under his guidance. And that really was a turning point in my life because instead of studying solids and solid state physics, I was studying living cells, and we're measuring currents in those cells. It was absolutely fascinating. So we developed a whole new technique, which at the time, we called the vibrating probe. It was basically an electrode that vibrated back and forth, and we used a lock-in amplifier to measure the voltage between the 2 points. And if current is flowing in a medium, that will generate a voltage in the medium, and it's very small because you're in seawater. It's very conductive, so the voltage you generate is really in the nanovolt, 10 to the minus ninth volt range. It's very small, but with some fancy electronics and averaging many measurements, you can get down into those regions, and we did. And we measured currents around the single fucoid egg, a Pelvicia egg, and that's how I got into it. And it was ever since then, I've been looking at those kinds of things, fields and currents in cells and their response to imposed fields, because that's so critical for development of our organisms. So it's really been fun.
So when you first got into this research, you developed the vibrating probe with Lionel
Yeah.
Jaffe as well. What was the next step? Like, what was the significance of bioelectrics back then? And one, I guess, was it even called bioelectrics, the field?
Not really. Bioelectric was a term that was coined much later. There, it was simply called ionic currents in development. We had a whole book that we wrote called Ionic Currents in Development, and that was about we just referred to, see, when people talked about currents, they thought about electron movement in wires. And, of course, we aren't talking about that. We're talking about sodium, potassium, chloride movements through cells. So that's why we emphasize the ionic current aspect, because that's a whole new way of thinking of current with ions carrying it. Yeah, that's the whole way we started describing it. And it was kind of new. I think that it was fairly new. People didn't quite know what to make of it at the time because of, they didn't think of cells as driving currents through themselves. It was a whole new way of thinking. And, as Mike will attest, it still is. somewhat difficult to get people to realize that's what's going on. So, but it's I think it's largely because it never quite made it into the biology literature, into the biology textbooks. They teach all kinds of things about genetics and physical chemistry and all kinds of biochemistry, but you don't get much biophysics taught in the biology realm. So even doctors growing up, they don't learn about this stuff until they get into cardiology and they realize that their heart is controlled by electrical voltages and currents, and they start realizing how important it is then. But before that, in their basic training, they just don't hear much about it, and that's unfortunate, because I think it's the basis of life is bioelectric, and it's just not completely understood and taught that way. But when you think about it, every single aspect, every organ in your body has a voltage across it, across the outer boundary, the epidermis, or the outer layer of the organ. The kidney does. It has an outer envelope, the liver. And there's a voltage across each one, and that voltage is important for the function of that organ. So we are electric beings, that's for sure. And it's something that's very, very slow to be realized by the medical community or anybody else, really.
So,
Yeah,
this is one. Go on, Mike.
Well, I was just going to say that I came across the first paper on the vibrating probe. I first read it in 1987, and
Right.
I was like, oh my God, it was obvious that this was a breakthrough technology, because for the first time we were able to see this living pattern of electrical activity around and spatialized too, around all of these things. Can you just mention real quick the range of things that it's since been applied to? I mean, it's been applied to a million different things, right?
Oh, absolutely. Yeah, it's been picked up by about 20 to 30 scientists around the world.
Yeah.
And they've used it to measure the currents through germinating pollen tubes, through the gastrulus stage of many embryos. We used it to measure the current through a blastomere. Just a mouse blastomere has current going in apical end and out the basal end, a constant steady current. We measured that many years ago, but it's been shown that in the chick embryo and in the frog embryo, during gastrulation, the currents are coming out of the neural tube region, and those currents are generating a voltage within the embryo itself that's very important for its polarity. And if you disrupt those voltages, literally change the voltage inside the embryo, you'll completely mess up development. They don't develop properly. You get all kinds of monsters created. And that's one of the things that Mike's been working on as well, looking at the molecular aspects of those pumps and leaks to see how that's involved in pattern formation. And it's critical, absolutely critical. So the probe is used quite a bit. It's not currently being used by many people. I don't know of any current labs that are using it every day, but there's about 100 or so papers out there showing different organisms that has been used to probe to determine the current patterns. Yeah.
When you first got into bioelectricity and started recognizing that our cells are electric, what was the thinking in the field around what the significance of this is? What was the main research interest around what's going to happen with understanding that our cells are electric?
Well, the first thing we did when we actually measured the current in the fucoid egg, it's a seaweed egg that germinates a rhizoid at one end. So it starts out perfectly symmetrical sphere, and it has the capability of growing anywhere on its surface, and it grows by secreting material that then softens the wall. So it grows by pressure, you see. It builds up pressure on the wall, and as the wall gets softer, it expands, and that's how it elongates. So it turns out that with this particular organism, it looks at the cues around it, mainly light and temperature, and it grows to the dark, so it goes to form a holdfast to it sticks to a rock, and it can grow up from that. It turns out that that current is critical for determining the axis of growth, and you can actually change that by changing the current flow. So normally it controls the current by the light being absorbed by photoreceptors, which then change the permeability of channels in that area and open up a calcium channel that lets calcium in, and that stimulates secretion. But if you then take that same cell and you put an electric field in a different direction, it'll grow towards that electric field. So it listens to what's out there, and if it's light as the main factor, it'll listen to that. But if it's an electric field, that's the main factor, it'll listen to that. So it depends on the different environmental variables that it cares about to make its decision. And I think that's the case with many cells in the body. When they are developing, they're in an environment. They're surrounded by other cells, and they get information from that. And often that information is electrical, so that they have a current nearby or a voltage gradient nearby that it affects the way that their ion channels and pumps are oriented or localized on their plasma membrane, and that will then in turn generate a little current in their through them that will polarize them. So basically, the electric fields that any cell experiences will certainly influence the way it grows in both direction and its differentiation. The genes that are turned on, and that's one of the things that Mike has been finding, that genetics is listening to the electrical signals and basically making decisions based on these external inputs. Another thing that's not in the literature. So not yet.
So this is a question I have for both you and Mike then. Mike, when you said when you first read about the vibrating probe, your head was just like, oh my gosh. And obviously this field clearly still remains underrated, even though there's a bunch of fascinating research happening. Why do you think biophysics or bioelectricity has not made its way into the textbook or still remains so underrated?
Rich, you want to go first?
No, I think you should answer this one.
Okay. So here are my thoughts. One of the things that make biochemistry and genetics a lot easier is that you can study them in the dead state. So you can fractionate your cell. You can isolate the DNA, the RNA, the proteins, the lipids, the sugars. You just spread this stuff out, and then you can analyze them. And so the amazing thing about bioelectricity is that it's fundamentally, I mean, as a number of people have said, it's the spark of life. It's only there when the system is alive and functional. So that means that it had to really wait for technologies like what Rich created and various things since then, the fluorescent dyes that we've used and so on, before you could actually get these data. Molecular biologists and biochemists have been at this for how many decades, from long before that, because you can break up your cell and that's good enough. So that's the first thing, is I think it's more difficult, and the technology needed more time to catch up. Also, I think as Rich said, you kind of need a different mindset for this. It's not as simple as the kinds of things that you normally would learn in a biology and chemistry kind of to really comprehend it. And I don't know if Rich agrees, but to me, not only do you need physics, but you also need a lot of what's now basically neuroscience. You need a lot of computation to really understand what's going on. You need to understand computations. It's a very sort of fundamentally interdisciplinary field, and I think that's harder.
Yeah, definitely. I agree with you completely. And again, I think that a lot of the problem is that our classical education divides things up, and a biology student doesn't get enough physics to really understand electricity and magnetism to a depth that makes them comfortable with it. They typically get maybe one semester, at the most 2 semesters of physics, and it's just enough to hate it and not to enjoy it and really have it become a tool. So they're afraid of it, and because of that, they don't really go beyond it into the biophysics needed to really appreciate the ion currents and those kinds of things. I think that's a large part of it, and I don't know how to fix that, other than having that be changed in the mindset of our education system to get more physics to all the biologists out there and all the pre-med.
Yeah, but it's in the Gilbert Developmental Biology textbook has 2 bioelectric stories in it now, so it's
super.
It's getting there. Yeah.
Yeah, that's great to hear.
Have you noticed then that most of the scientists who are looking into the field, do they also have a bit of a physics background?
Yes, in general, that's the case. When thinking of the ones that I know of that have taken up, have used the vibrating probe, for example, those guys all had to have some pretty good feeling about physics to understand how to interpret the data and that sort of thing. I think that is true.
I mean, there's another way in which people get dragged kicking and screaming into this field, which is that somebody who does regular genetics will do a screen of some sort and they'll get their mutant and they say, oh, this is fantastic. This is the thing I need to study. And then they find out it's an ion channel. And I was
exactly.
like, what? And so now there's been like this, amazing work by, Emily Bates and
yes.
In the Harris lab at Harvard, where they do these purely genetic screens where nobody was out to find anything bioelectric, but then you find that oh my God, it's a channel, and so now I need to understand how this voltage change gives rise to an altered wing or size control or whatever. And there's been there's probably almost 3 dozen good developmental channelopathies now that people have studied that bring them into the field, even though they weren't really trying to study that.
Exactly. That's a great way to get in, to be pulled in unsuspectingly. I love that. I love it.
Yeah, I mean, I would often when I give talks to those kind of audiences, I would say at the beginning, how many of you have done either a microarray or RNA-seq for your, and they said, well, so how many of you have found ion channels in your top list? Oh, absolutely. The first 10 things were ion channels. And they said, well, what did you do? Oh, well, we skipped them and went to the transcription factors, of course.
Yeah, right. Exactly.
Right. And so because the tools were there, and so that's one of the things that we worked on pretty hard, is to develop tools and protocols that we would share. And then I would say, okay, so everybody who had that, here's a how-to manual, and here's some reagents and whatnot. Now that you can, now that we can study these things.
Yeah, fantastic.
Now, is the reason they skip from the ion channels to say the transcription factors or other factors, is that because they don't recognize the significance of the ion channels? Like they don't at that point recognize that it's doing much, or they just don't have any knowledge of it?
Exactly. That's the bottom line, is they don't have that basis of every cell has a transcellular current. They don't think about that. They aren't taught that. It's farthest from their mind, but it's absolutely true. We haven't found a cell yet who doesn't have a transcellular current, which is intimately involved in its function. Think of your epithelial cells in your gut. I mean, they're polarized. They have sodium channels on the apical side, potassium on the basal. They have current. They're generating a voltage across themselves. That voltage is intricately involved in the digestion, for crying out loud. So things as basic as eating and digesting requires these transcellular currents, but they don't know about that. It's not part of their education.
Well, and there's also a subtlety here, which is that when you have a transcription factor, you can tell a story about a gene that does a particular thing, and that's a very popular story. But what you find is a popular kind of story. But what you find with these ion channels is that it isn't actually the ion channel gene or protein that's doing it. It's the physiological state that ends up being the critical factor. And you can get
Yeah,
the same state by different channels, and you can get multiple states from the same channel, depending on whether it's open or closed.
exactly.
Right. So seeing physiological patterns as a causal factor is not what people are used to, you know?
That's right.
Right. And then it goes back to what Rich was saying before about these different fields. So I remember in 2007 we had this paper on there was a particular proton pump that was required for tail regeneration in tadpoles, and then we showed that if you get rid of that pump, you can rescue it back with a yeast proton pump that basically has no sequence homology but does the same physiological job, right? So
Yeah.
we send this paper out and we get 2 reviewers, and the first reviewer says, oh, and and of course we had imaged the voltage and all the stuff. This was Danny Adams was the first
Yeah.
author on this, right? And so the first reviewer says, oh, good, you found the gene for tail regeneration. Get rid of all this voltage stuff. You don't need it. You got the gene for tail regeneration. This other stuff is junk. And then guess what the other reviewer says? Oh, well, clearly the gene doesn't matter because you've replaced it by this thing from yeast. So get rid of all of that stuff and just keep the right?
Unbelievable.
Right. Yes.
Yeah.
And we wrote to the editor saying, so what do we do? And the editor said, I don't know, but to make it shorter. So
Yeah. Make it shorter.
make it shorter.
Oh,
So,
that's terrible.
yeah.
Yeah.
So that's that's the thing, right? You have to think a little bit differently. Like it's not the gene, it's the and then it gets progressively worse after that, because then you end up it's patterns. It's actually the voltage patterns that determine what
Oh,
happens next.
exactly. Yeah. Even more difficult to explain.
Fascinating. Okay, so I'd love to understand a bit of the progression of the field and the research interest from, say, the 1970s up till now. Maybe a good way to do this would be a comparison, or you could just, so one way could be maybe you can talk about what was the predominant thinking in the field in the 1970s and 80s and 90s, or you could also tell me how it compares to today and well, how the thinking was different back then.
Wow. You're asking a
Well,
lot there.
and if you don't mind, I actually want to make it even harder, because I was going to and you can answer that later or separately or whatever, but I'm even interested in going back further before that, right? Because there was some great work in the 20s and 30s and 40s, and so
Oh,
anything you
yeah.
want to say about kind of all of that stuff and grow, going forward, we'd love to hear it.
Oh, that's absolutely right. There, yeah. If you go back to the early 1900s, where people like Burr and oh gosh, there's a variety of very well-known biologists who are actually looking at these kinds of things, measuring voltages with cotton wicks soaked in saline and putting it on different regions of embryos. And that work was almost lost. It was, it was really groundbreaking, and so unique that I think that it was just not quite appreciated. And the main reason, I believe, is that in the 1950s, the whole DNA story came out. So the molecular biology took over, and the 1930s, 1920s, the whole work that was a body of work measuring electric voltages and showing that there are fields in these embryos was sort of forgotten in many respects. But that was sad, because yeah, certainly the molecular revolution, the DNA revolution, was huge, and we thought that it would explain everything. We now know that it doesn't, but it certainly made us believe that we could understand life by knowing the sequence of our DNA. And I think even molecular biologists now agree that that doesn't tell you how things work, that it gives us some idea of how proteins are made and what the structure of proteins are, but it doesn't really, it doesn't help us understand life completely. And that's where Mike's stuff is really much more important. But in looking at the evolution, The molecular revolution pretty much, in my opinion, completely overshadowed all the physiology. The developmental physiology that was going on just went away and was not even looked at anymore. I think when Lionel's generation came in, he was fairly unique. I don't really know of a lot of comrades for him that really he could work with. He was a very unique person, and he started looking again at these electrical currents in developing embryos and in plants, mainly in these pelvicia, and he sort of raised the awareness again of this, but it never really took off to a huge extent. He didn't have that many followers. His students all followed him. Ken Robinson and Rich Borgens and myself all continued doing his work. And we had a few other, maybe a dozen other scientists who started using the vibrating probe in Germany, in France, in Italy, and made some progress in the field, Canada. But when that's sort of in the 70s and 80s, and in the 90s and in 2000 or so, is when I sort of found the role of ultra high voltage, ultra short fields in cell signaling. So that's a whole different area of bioelectricity that I've been exploring for the past decade or so, 2 decades really. And that's another aspect of bioelectricity that is just fascinating. That so I started out measuring nanovolts, 10 to the minus 9th volts, and now I'm applying a 10 million times larger field of 50 kilovolts or 20 kilovolts. So, but in a way that's still physiological. So it's a way that still is no heat, that is fractions of a joule, but it communicates to the cell in a way that gives it a message. And the message is to undergo regulated cell death and to start doing something that it would normally do at the end of its useful life, but start doing it now. So it's a fascinating area that I've been exploring and really enjoying.
Can you talk a bit more about that? About the ultra high voltage field? What's
Sure.
there?
Yeah, that's what's called these nanosecond voltage pulses. So the whole idea is that basically something that was found back in the 50s, actually, 1954 was when the first discovery of a pulsed electric field could permeabilize cells. It was called electroporation, still is called that. And what happens is you make electric field that's strong enough to push water molecules, which are waters in normal out in reality. Water is a dipole. It's H2O, 2 protons and 1 oxygen, and that has a net positive charge. So when you put a voltage, it feels the voltage and it moves along the field. And if you put up to about 400 millivolts, maybe half a volt, across a bilayer, a lipid bilayer, which all of our membranes are made of, if you put a voltage across that of about half a volt, it's strong enough to push water into that lipid and form a water-filled pore. And that is permeabilization. So now this lipid bilayer, which normally separates inside from out, has a little leak. And that leak would let ions pass through it if there's a gradient of force. So what we're finding is that normally if you put a typical cell is maybe 10 microns, between 10 and 100 microns. And if you put about 1,000 volts per centimeter, 1 kV per centimeter across that, that gives enough voltage across the membrane to permeabilize. What we found with higher fields, if you want to permeabilize the organelles, like the mitochondria, the ER, now that 1 kV is not enough, because now you're looking at a much smaller distance across that organelle. So you need a larger field to generate the same half a volt across that membrane, because the membranes are much closer together. So now we have to have 20 or 30 kV per centimeter. And that's possible only with ultra short pulses. So 1 kV per centimeter you can use with a microsecond. It's no problem. It doesn't generate enough joules or heat to cause too much heating. But if you go up tenfold, up to 20 kV per centimeter or 20-fold, now that at 1 microsecond pulse, that would be way too much heat. It would boil the cell. So the joules that you're introducing is too much. So you have to shorten it. So instead of a microsecond, you can do a nanosecond, which is 1/1000th of that, or even 10 nanoseconds or 100 nanoseconds. As long as you're less than a microsecond, you can get down and introduce it. And now the trick now is not only do you permeabilize the plasma membrane, but you permeabilize the mitochondria. And that's important because mitochondria function by generating 200 millivolts inside negative across their inner membrane. That voltage is critical for making ATP from ADP. So it adds a phosphate onto ADP, and that phosphate is added in part with the electric field. There's also a proton gradient that's important. So it's a combination of a proton gradient, pH, and electric field that make that ATP. But if you permeabilize the mitochondria, make it leaky, so it can no longer generate a voltage gradient, you don't make any ATP. That's been shown. And that's the magic of our energy, is that we not only permeabilize the outer membrane, we permeabilize the mitochondria. The water pores in the mitochondria make it swell. Water moves in because of the gradient. It moves in and the mitochondria swell and they stop producing ATP, and that
Did you lose Rich as well, Mike?
I did, yes. Sorry, Rich.
Yeah.
Hang on a second. Your sound is off. Rich. Oh, I don't think he can hear us either, huh?
Yeah, I don't think so. Rich,
Rich,
can you hear us?
We can't hear you.
Yeah, I think he.
Say something.
Oh, that's better.
go.
Now I can hear you.
You're back. Yeah.
Okay.
We lost about maybe 15 seconds or so.
Oh, I'm sorry about that. It must have been my headphones. Yeah. So where did I stop coming across?
You were talking about how the mitochondria would basically lose the energy in the ATP.
Yes.
You can permeabilize it.
Right. And so that when that happens, not only does the cell lose its energy source of ATP, but now messages typically are released by the mitochondria, for example, Cytochrome C, which is one of the messages required to initiate regulated cell death. So it starts the pathway that all cells have when they find that they're not functioning well. They become leaky or they had been damaged. And that pathway is what normally occurs. They release the cytochrome C. They make a message go from the ER to the plasma membrane, calreticulin. And then when the calreticulin is expressed on the surface of the cell, dendritic cells that are always looking around for problems, they bind to that, and that's a message to phagocytose that cell. So it's a way for our body to go after cells that are damaged, clean them up, and rescue any problems that are happening.
Interesting. So does this have significance for maybe cancer research and killing off cancer cells as well?
Exactly. And we've been using it to treat different cancers. So in a mouse, for example, we're able to make mouse models of all sorts of cancer, from pancreatic cancer to basal cell, carcinoma, to a whole variety. And in a mouse, if you can make a tumor, we treat it with putting our electrodes around the tumor. We simply give it a few pulses, and it dries up and goes away. In literally a couple of weeks, the tumor is completely gone, and it never reoccurs. And when I found that with these nanosecond pulses, I was absolutely blown away. And that happened in 2007, and ever since that, I've been working on that. And we now have Pulse Biosciences, where I'm working. It has generated a machine, a pulse generator for this. It's been approved for use now for treating benign skin lesions. And also now we've just had an electrode, a percutaneous electrode approved that we're using to treat thyroid nodules by putting the electrode into that, and it causes the nodule to shrink and completely go away. So we're starting to use it to go after problems in our medical field.
Wow. And is there any damage to the good cells, like in chemotherapy you might have?
Yes, there can be. You just have to be careful because every cell will respond to these fields for sure. That's what it's a good thing and it's a bad thing, because it's a biophysical phenomenon. You can't prevent getting holes in membranes when you pulse with these fields. It's its physical property of the lipid bilayer. So when we apply it, we're always careful to try to limit the energy to the tumor so we don't damage other cells. But the fact is that in the skin, for example, we've done a lot of treatment of skin lesions, and skin cells are pretty good about recovering. And for example, when we treat a wart, which is embedded, we'll typically get a few of the skin cells damaged as well. But skin is amazing for regeneration. As you know, when you cut yourself, it heals so beautifully and recovers. So that skin can regenerate, and we damage a few of those skin cells, and it's not a problem. But the main thing is we get rid of the wart. So that's typically how it works.
Going back a little bit to the progression you mentioned, so early 1900s, there were people doing some experiments with cotton wicks and realized that, okay, there might be electrical forces in our body, but a lot of that research was lost. Then
Yes.
1950s, you said, because of the whole DNA research, the entire field, everyone was focused on that.
Definitely.
And then Lionel Jaffe came along, and you and Ken Robinson, and you had the Vibrating Probe, which made it much easier for people to detect these electrical signals in the body. Around that time, when people started detecting electrical signals, I'm curious, when did they start realizing that these electrical signals play a very significant role beyond the DNA? In other words, first you discover that, okay, our body is electric, but when did they start realizing what the significance of the electrical signals in the body could be?
Well, that's a great question. As I think about it, the field really was concentrated for a long time on this technique called patch clamp. And this is what electrophysiologists use to study ion channels and the way the channels operate, the gating of channels and that sort of thing. And I think when the patch clamp technique was invented, that was again around, when was that? I would say that was in the 1980s, between 75 and 80. That really made electrophysiology all concentrate on that technique. And that's how they decide how they were able to explore ion channel opening and closing, the timing of it, and that sort of thing. But when you think about the polarity or development of the asymmetry in channel activity, it's really hard to put your finger on that as a phenomenon that people really appreciate. I think it's really the work that Mike has done that has made more progress in people understanding the importance of that. You know, one of the things that we showed over the years in the 1980s and 90s were currents, leaving growing limb bud stumps in regenerating limb buds and in tails and those kinds of things. Those currents were certainly published and people recognized that they were happening. But I still think that this whole phenomenon of developmental currents and voltages is something that people are still trying to get their head around. Would you agree, Mike?
Yeah, yeah. I mean, so there's a few. I was just looking up, right? The earliest paper that I have, I mean, you could go back to Volta and so on, but the earliest
Sure.
developmental paper I have is 1892 from Wilhelm Roux.
Right.
Right, and it's 1892, and he's writing in German about the effect of the electric fields on chicken embryos and things like this.
Exactly. Yeah.
And then you had well, like one of my old favorites is Burr, so Harold Burr. So this is like 30s and 40s, and this
Oh
guy
yeah.
just right. I mean, he basically had a good voltmeter. That's all he had, and
Right.
he went around measuring everything from embryos to maple trees to various psychiatric patients. I mean, just dozens and dozens of papers. And in his book, he extremely prescient. I mean, he said things that he had no way of being certain of, but that in the end turned out to be quite correct.
Yeah.
He saw a lot of it. These pre-patterns and these like subtle voltage pre-patterns for, and then another guy I really liked his work is Cohn, Clarence Cohn in the 70s. So he showed that if you depolarize neurons, they re-enter mitosis. And right, and so he was really pushing the resting potential, this idea of resting potential.
Absolutely. Yeah.
Right.
And yeah.
Yeah. And then in 2000s, what we started doing was really tying this stuff to the molecular genetic downstream consequences. So like our first paper with Ken Robinson was on left-right asymmetry in the chicken. And the deal there was that there was this very important gene, sonic hedgehog, and everybody wanted to know about sonic hedgehog. Well, it turns out if you want to know why sonic hedgehog is turned on the left side and not on the right side, you have to know about the voltage gradient that precedes it. Otherwise, it's a big mystery.
Exactly. Yeah, no, that's fascinating. So really, it's all coming together when you look at the way you've integrated the molecular biology with the electrical field understanding, pH gradients, that sort of thing. Potassium ion currents, potassium pump location, so critical. It's amazing. Yeah.
Rich, do you want to talk a little bit about what was it like working with Lionel? Because I knew him a little bit, and I always thought he was a very interesting character. And you obviously knew him much better. What was that like in his lab in those days? What was it
It
like?
was amazing. Yeah, he was quite a character. I'd say not only had incredible physical intuition, where he just had a way of thinking about these things that I hadn't encountered before him. But he also was very much into politics and very strong in his political beliefs. He was very liberal, and almost every afternoon when we had our coffee or any time to sit and chat, it would be about politics. It was amazing. So he and luckily Ken Robinson and myself were also very liberal, so we were able to get along pretty well. But I'd say the thing about being in his lab is that he worked very hard He was there typically around 8 in the morning and would often stay till 8 or 9 at night. Had no problem staying late hours overnight to when we have an experiment that had to go 24 hours, he would share the nighttime with me. So I would take the first shift and he would come to take the second shift. He was a very hard worker and very fastidious. So when he started doing his work, first thing he does is clean everything. Everything gets removed that's not necessary. You only have the instruments you need. And he was very particular about that. And it was important because the stuff that we're doing, the developing the vibrating probe, required very fine work making the probe itself, which was a very fine needle that's with a platinum ball on the end of about 30 microns in diameter, which you make under a microscope. So you watch it grow in front of your eyes with electric field, to attract the platinum to the surface. So that is all pretty fine work. We typically would have Bach in the background, his piano concertos or his music to keep us calm while we're doing this fine work. But it was really a fun place to be. Tell you the truth, everyone that was in the lab really enjoyed being there.
Would you say that it's always interesting to me the motivation that great scientists have, and would you say that he and you and I guess the rest of you were you motivated by a specific outcome or capability or kind of an endpoint that you wanted to get to, or was it more just the curiosity of whatever showed up in the kind of the in the moment more sort of joy of the experiment? How did you guys see that?
Yeah, I think that it was the joy of the moment. There, none of us knew, didn't have an expectation of where we're going. This is all new. This whole thing of currents around cells, it was completely a new concept. We didn't know where it would take us. So that's why one person in the lab would put a field across the cell. Another person would try to measure the normal current. Ken was trying to figure out what ions were carrying the current. So he actually put the eggs in a metal screen separating one side from the other, and he used radioactive tracers to determine what's going in and what's coming out. So we had all these different approaches to understand the mechanism. It was fascinating place to be. It was all discovery of completely new things that no one had heard of before. So yeah, it was an exciting time. But we were there just to try to understand how it worked. What was carrying the current, why there was a current at all. What was driving that. What was, and that was fascinating. So we can actually show that the electric field could change the current direction, and that would actually polarize the cells. So you can actually make the cells grow in a certain direction by the electric field. And how does that work? What was going on there? And that we showed that it was affecting the calcium permeability, which then affected the chloride permeability. So it was a remarkable time. Still have great memories of it.
Mike, what was your impression of Lionel? You said you interacted with him briefly.
Yeah. I met him a few times. Also when he was at Woods Hole, I would go up there and meet with him. Yeah, definitely a kind of an iconoclast. He was always thinking in new directions, and wasn't particularly concerned with what anybody thought. Had all kinds of wild ideas. Yeah, played tennis. He was quite advanced in years, and he was still playing tennis. He complained to me about not being able to find enough partners at that point.
That's true.
Yeah.
I played tennis with him. I remember that. That was great. Yeah.
Yeah, really.
Mike, when you first entered the research and entered the field, what was your understanding of bioelectrics?
Well, I mean, first of all, by the time I went through by the time I had gone through undergrad and grad school, I had been reading what Rich and Lionel and Ken and Richard Borgens and those guys, you know, they were my heroes. Okay, all you guys were my heroes, and I've been reading those things. I ate and slept and dreamt all of this stuff, but I also knew it was pretty clear that at a certain early stage of the game, I better keep my mouth shut about all this, and so I, you know, I kind of I got my degree in genetics, not saying anything about any of this, straight up just learning the kind of the molecular genetics of chick development with Cliff Tabin and so on. But then during my postdoc, I started sliding into it because I figured that, you know, pretty soon, if I had my own lab, I would be able to really get at, really get at this. And around that time is when I started being able to meet you guys in person, and I would go to, I went to the Gordon conference, and then especially eventually I had some actual data to show. I was just so thrilled. I was so thrilled to meet everybody. They were helpful and they were excited, and which I found amazing, because all the years, I mean, you guys did not have it easy, to do all that work against the kind of the mainstream of the genetics and the molecular biology, and to hold on to that. That's what I remember. I remember the excitement about the field, like not at all diminished by all the years of hard work and arguing with people, and and having to break through with this stuff. And I just remember thinking that this is great, that if I was able to ever do this, the key thing to hold on to is that sense of wonder and that sense of excitement about the work, and not let all the difficulties of it sort of get you down. That's I was amazed at how welcoming they were to this new young kid who didn't know anything and was but was also excited about the field. Yeah, I had really good experiences there.
Yeah, and it was we were excited to see a molecular person come in with interest in our stuff, because we had just not seen much of that. So you were the first one to come along and show an interest and understanding the connection between the ion currents and the molecular biology, and that was fantastic. We were so happy to see that.
Yeah, I couldn't believe I mean, I just couldn't believe I got to that I got to meet all you guys in person. I would spend from I first ran into this field in 1986, and then from there I spent the next, and this was now before the internet. Okay, so there's no searching that you can do online. This is like in a dusty in a dusty basement of a library somewhere, Boston Public Library, trying to find these things. The catalog barely was functional, trying to get, and just finding all of these papers, and each one of these papers had citations to other papers, and you got to go chase them down, right? And so and you make photocopies, and then you sit there and you read all of this. So this is just that's literature that I was eating and dreaming of for years.
Phenomenal.
That's just so amazing to hear. Rich, maybe you can talk a bit about the criticism that the field received when you, Lionel, Ken were coming up with all this research.
Well, when you say criticism, I'm not sure that we had much problem getting it published. We always did experiments with good controls and a large data set, so that we published in good journal of cell biology, developmental biology, without very much problem. So I don't think that there was any criticism in that regard. I think the main question is why more people weren't excited by it, and why we didn't have a lot more followers than we did. And again, that was a molecular biology aspect. I think the most of the biologists out there found our stuff kind of intriguing, but didn't get excited by it as we were, and didn't quite appreciate the importance of it. And I think that's still the case. I really don't think that the importance, the role of these ion gradients and currents in development and in physiology is really appreciated by the bulk of the community of biology. Would you agree with that, Mike?
Yeah, I mean, I think it's gotten a lot better. I think once again, we're in. I mean, people publish this stuff now in Cell and Development and whatnot. So I think we're sort of back there, but still, a lot of people have never heard of it. So that's always an issue.
Right.
And there's kind of there's kind of a sister field to this, which has had a similar journey, which is biomechanics. All the
Yes.
physical forces, the stresses, the strains. So there were all these people in the, in the past decades, Lev Belousov and all these other people that were talking about the importance of all these things and writing these papers, and they're completely ignored until some of the technology came along, and people like Don Ingber and other folks who made molecular tools where you can now make high resolution quantitative models of what the actual physical forces are doing, not necessarily the genes, but the actual physical forces, and the fact
Yes.
that if you culture cells on a square peg versus a triangular peg, you get different genes turned on and this kind of thing. So I think they went through the same sort of thing, and again, very difficult time until the technology showed up. And I think technology in these cases. So two things make a huge difference: the technology, and then what you might call killer apps, right, in the computer science field. Something that, okay, now you can see, you can do something now that you couldn't do before, and that gets people's attention, and then the young people sort of come up with knowing what it's about.
Exactly.
What's
Yeah.
the technology that you think is missing now or would be helpful to advance the research further? Question for both of you. Mike, maybe you can go first.
Okay. Well, I think the biggest thing that's missing, there's a few things, but one of the biggest things that's missing now is the physiomics data sets. So we have these massive, just think about trying to do molecular biology before we had genomic data and transcriptomic data and all this stuff. What we still don't know is if you wanted to know in some model system, some organ under some disease state or at some stage of development, what is the bioelectric pattern? We only have like a tiny number of these things mapped out. And Rich has done a ton of it with the vibrating probe. We've done some with the voltage dyes. But in the grand scheme of things, there's very little bulk data available. And that's pretty critical because, modern tools like AI and other techniques require a lot of data to really extract the signatures and functional target endpoints and so on. So that's one of the things that we're focused on now, is trying to develop technologies where in some sort of high throughput way, we can actually generate these data sets so that we actually know what the native states are for these things. What do you think, Rich?
Yeah, no, I think you're absolutely right. I remember that beautiful paper that you guys came out with when you labeled the frog embryo with the dye that measured membrane potential in the different regions of the embryo, and showed that actually got a visual picture of the gradient of potential in the blastomere stage of the frog embryo. And that is what we need. We need to have a visualization of the electrical properties that we're seeing. And it's so hard to do because the organisms that we want to study are multicellular, thousands and thousands of cells, but those cells are working together in a very intricate way. The ideal would be to be able to image the field, the whole electric field everywhere in that organism, and that would be a dream. It's not obvious to me how to do that, but I'm sure that one day we will have some molecules that will show the gradient in a way that we can image them. And there's quite a few different tools that are working towards that. This whole optogenetics business where you control gene activity with light. Be nice to be able to reverse that and show have light come out with the bioelectric activity. That would be fantastic. I hope that happens someday. I don't see it on the horizon yet, but I wouldn't be surprised.
Well, we've so we and others have now are generating transgenic mice where every cell is expressing one of these optogenetic voltage reporter proteins.
Okay.
So I think I think in the next couple of years we're going to have model systems where every cell is advertising its electric state, and then we'll be able to start. Again, you still have the issue of it's hard to see through tissue, right? And so what's going on? You still have all that, but at least the cells will be sort of trying to tell you what their voltage is as much as you can see them.
Exactly, that would be fabulous if we can get that to work. I love it.
Now, maybe this is a stupid question, but do different humans have different electrical states around the cells?
Different humans.
As in you and me and Mike, are our electrical states around tissues and organs and cells going to be different, or do all cells, like normal cells, have the same type?
I think it would be generally be the same. I guess it depends on the disease state. So clearly, if I would say that as you get older, I know for a fact that the patterns and the voltages get lower. So I've shown that in a skin wound, for example, as you age, the amount of current flowing, the wound current, goes down. You just don't have quite as much energy, so to speak, to drive the current. So I think you would find a difference as a function of age and any kind of disease state, for sure. In general, a healthy individual will have very similar patterns than not. It really depends on what you're looking at, but typically you'll find a voltage across every epithelium, every organ, and that voltage will drive currents depending on what that organ is doing. Yeah.
I mean, there's also there have been stories in the cancer field about sodium versus potassium-rich diets, and this, right, and so one can hypothesize that if what we're talking about is this kind of like a change in resting potential due to sodium and so on, you might expect, right, people with different diets to actually have shifted VMEMs that would predispose you to different different conditions like cancer and so on.
Absolutely. I mean, it's been shown pretty well that depolarization is required for a cancer cell to be very active. They're typically, when you look at their membrane potential, they're more depolarized than a healthy cell. So that obviously you want to avoid that to avoid a cancer state. So that's absolutely right. Yeah.
Last question for you, Rich. I know we are over time. What would you like to see developed in the field going forward?
Wow, I think we just answered that. I think this whole idea of getting cells to tell us what's going on in their electrical activity would be fantastic. So this thing that Mike just mentioned, having a reporter gene that expressing a signal such as a light emission or a fluorescence that we can detect, that would tell us if that cell is depolarized or hyperpolarized, or if there's actually a current going through it. That would be fabulous. Then we can have an image of the whole electrical property of the organism in different areas of the organism. That would be fantastic.
Okay. Well, thank you so much, Mike. Do you want to add anything? Any last questions?
No, this was great, Rich. I just want to say thank you so much for taking this time with us. It was awesome to get your thoughts on everything. Yeah, thank you.
My pleasure. It was fun to talk about. I'll be glad to talk anytime.
Thank you so much. Yeah, this was wonderful.
All right, guys.
Thanks, Rich. I'll see you in the Bioelectricity journal. Plenty more.
Oh, yeah, absolutely. Yeah, okay, good to meet you. Bye-bye, Aastha.